let's do some practice with the dihybrid crosses because that's where you guys were struggling earlier today. So, to begin, I've set up a 4x4 dihybrid Punnett square here, and we need to figure out what the genotypes of our parents are going to be. So in your homework and the practice problems we've done, most of the time you're either given the genotypes already or you're given it in the form of a word problem, meaning mom is heterozygous for this trait and homozygous dominant for this trait, and dad is heterozygous for both traits, and you have to figure it out yourselves. So for this, let's just say we have them already, and let's make them here. So let's say that one of them is going to be, let's use T's and G's. Let's say T is tall, dominant, and G capital is green. So let's make one parent homozygous dominant tall and heterozygous green. Now let's try this. Let's make another parent heterozygous tall and yellow. Okay, so this is mom here and this is dad here. What we want to do is find out, if you remember the steps involved, we need to figure out what the gametes are going to be for these parents. Meaning, what sperm and egg can I make with these parents? And if you recall, we discussed a process called FOIL, right? So let me show you how we do that again really quickly. Let's make these guys a little bigger so we have an easier time seeing them. 20, there we go. Let's make that 22. Okay, let's bring them a little closer together. Let's space these guys out apart a little bit so we have easier time doing our FOIL. All right, so remember what FOIL means? It means first, outside, inside, last. Let's see what we can do here. Just a second here. Configuration, sorry, I'm just setting up this. Let's make this one red. Okay, that looks good. All right. Wait, hold on, that wasn't correct. We want to switch to pane. Yep, pane 1 is going to be red. Okay, so if we do FOIL, remember you start with the inside first, or sorry, the first ones first. So these are going to be our first ones here, big T and big G. So that means one possible gamete would be big T and big G. And then you do outside, right? So outside is big T, and then the other one outside is little g. And then FO, so first inside, or outside rather, and then you do inside, big T, big G, inside, and then last, the last T and the last G. So first, outsides, insides, and last. There we go. And when we combine them, these are what our possible genotypes are. Right? If we go down here, if I do big T and big G, I get big T and big G. That's one possible gamete. Okay? When I combine the first, big T meets big G, and there we go. That's one possible gamete. Sperm or egg, you pick. All right? Next, if we combine the outsides, big T on the outside with little g on the outside, would make big T, little g. That's another one. Okay. I could also, if I do inside, big T on the inside with big G, makes big T, big G. Okay. And if we do outsides, big T meets with little g. So big T, little g. There we go. All right, so that's how we did the first one. Now we're going to do the second one. All right, so again, we're going to FOIL this one too. Let's use a different colored pen for this one, okay? So let's make this one pen 2, and let's make pen 2 blue. And go. And pen 2 is going to be control 2, so control 2. All right, so again, first, boom, outsides, boom, insides, boom, and last, boom. All right, so when we make these gametes, we get first big T, little g. All right, sorry, little g. We then get outside big T, little g. We then go inside little t, little g. And then we do, let's go back, make some space here. And then we do little t, last, little t, little g. Okay, so those are our 4 gametes. Now, we need to plug in our gametes to our Punnett square. So, to do that, what did I do here? Oh, I made them all weird. Uh-oh, there we go. Okay, so now we're going to plug these in here. So I'm going to use my mom and dad. Let's make mom here. All right, so big T, big G, we're going to put up here. Big G. Okay. Big T, little g here. Okay. And then big T, big G again. Big T, big G. And then big T, little g again. Big T, little g. Okay. And then we're going to put dad on the side here. And dad we know is blue. So let's make this a little smaller so we can see what's going on. Oh no, that doesn't work. Let's keep it like this then. All right. All right, so there. All right, so we've created our gametes. I showed you the foil. Let's do this one more time. Control 1. Darn it. All right. Hold on, guys. All right, there we go. Let's bring these up here, make them a little smaller then. There. Now we can do it. All right. So then dad, we're going to put on the side here. Sorry about that. Dad is going to be big T. Oops, sorry, that's a big T. Little g. And then we have a big T, little g. Okay. And then we have a little t, little g. And then another little t, little g here. Okay. And those are our dad's gametes. So we've got mom here and dad here. And then we just have to cross them. So when we do that, we know we're going to get a big T and a little g from dad in this box. Okay. We can put a big T here and a little g here. And we know we're going to get the same thing in every box from this sperm that we got from dad. There we go. Big T. That's a big T, by the way. And a little g. Okay, and we're going to get this for every box because this sperm could fertilize any of these eggs. Okay, and then one more here. Big T, little g. Okay, and now we can do moms. All right, mom, we get a big T and a big G from. So big T, big G. Big T, little g. Okay, big T, big G. And a big T, little g. Okay, we've completed our first row. If we look at what the genotypes for these guys are, they are all, well, they're 50-50 because you see that we have 2 different types of gametes for mom here, okay? So this first one, genotype would be homozygous tall and heterozygous green, homozygous tall and homozygous yellow, recessive, and then these 2 are the same as the first 2, right? Okay. So let's complete our square. If we do that, we know we're going to get a big T and a big G from mom in each one. Big T and big G from mom. Big T and big G from mom. Big T and big G from mom. And then this one is a big T and a little t. or a big T and a little g. This one is going to be a big T and a little g. This one again is a big T and a little g. And then this one, big T, big G. Let's continue. Big T, big G, big T, big G, big T, big G, and this one, big T, little g, across the board. So that would be big T, little g, big T, little g, big T, little g. All right, now let's do Dad. We got a big T and a little g from Dad across the way, everyone here. Big T, little g, big T, little g, big T, little g, big T, little g. Ooh, that's an ugly one, huh? All right, and this Dad gave this whole row a little t and a little g. I'm supposed to put the little t's on the inside. Messed that up. I done goofed, guys. I done goofed, but be gentle. This is my first time using these tools. So it's not going to be pretty, but it's going to be done. All right, little t, little g. And this one, the same. Dad's, this sperm is the same as the others, so it's going to be the same. Little t, little g, little t, little g, little t, little g, little t, little g. Okay, there, we finally did it. That is our entire Punnett square. Wow, that was tougher than I thought it would be. So now let's look at ratios. If we want to look at these ratios, we have a certain amount of genotypes here, and we need to put all the like genotypes with like. So we can count how many we have that are the same, and we can just put a little dot for each one that matches. So this is homozygous tall, heterozygous green. Let's put a dot for every time we see that, okay? Come on. Okay, boom. Homozygous tall, whoop, that doesn't match. Homozygous tall, heterozygous green, that's a match, so let's put a little dot there. Okay. Homozygous tall, heterozygous green, little dot there. Homozygous tall, nope, oop. Okay, homozygous tall, heterozygous green, and there we go. And then none of these are going to be homozygous tall, so we can stop there. So we know that of all the T, the big T's, and the heterozygous G's, we have 1, 2, 3, 4. Okay, so if I were to write this, I would have, if we open, let's open a new one so that we don't have to mess with this one. Let's create a new document here. Okay. All right, let's put this one over here so we can bounce back and forth. Come on, don't be a poop. There we go. Okay. So we know here that we have big T, little g's. We also know that we have, control 1, this other one, big T's, little g's. So let's make a mark for every time we see that. Okay, and we see it there. And we see it here. And we see it here. And is that it? Yep, that's good. Okay, so then we know that we have big T's, little g's, and there are 4. So we got 4 of those. We got 4 of those. And the other genotype that we see here are heterozygous T's and heterozygous G's. So let's make that a different color. Let's make that one. Configuration. Let's make, okay, let's make pen 4 different. Let's make pen 4, what do you think? Pink? Yeah, I like pink. Let's do that. Okay. So, let's do 3. We know that heterozygous for T's and G's will make green. So there's 1, and there's 1, and there's 1, and there's 1. Let's write it over here. Okay, so heterozygous for all. And guess what? We counted 4 of them, just like before. And we see our last genotype is heterozygous T's and homozygous G recessive. So let's do that. And guess what? How many do you think we're going to have of that? Well, let's count. We have 1, we have 2, we have 3, and we have 4. What do you know, guys? It all matches. 4. Okay, so we have, you ready for this? A 4 to 4 to 4 to 4 ratio, or if we simplify it, a 1 to 1 to 1 to 1 ratio. Ah, there we go. Now what about the phenotype? That was the genotype ratio because we have 4 different types of genotypes and there are 4 of each kind. So that gives us this. But our phenotype is going to be different. Phenotypes, remember, are what these things look like. So we know that a capital T means you're tall. Do they all have a capital T? Yep, they sure do. So we know all of the offspring are going to be tall. But are they all going to be the same color? Let's take a look. If I have a big G, that's the dominant trait, and green is dominant. So if there's a big G, it has to be green. So let's see, how many are tall and green? Well, tall and green, tall and green. So 1, 2, 3, 4, 5, 6, 7, 8. Because there's big T's and big G's in these 8 boxes, okay? So we know that there will be 8 tall green plants. If you're not tall or green, since we can't be short because we can only get a tall allele from mom here, they all have to be tall. So you know you're not going to have any short. So the only other option is tall and, well, if you're not green, you're going to be yellow. All right, so how many do we have there? We have tall and yellow, tall and yellow, tall and yellow, tall and yellow, and then there's another 4. So 4 and 4 makes another 8. Okay, so this would be phenotypically an 8 to 8 ratio, but you know you can simplify that back down to a 1 to 1 ratio. Half of them are tall and green and half of them are tall and yellow. All right. I hope this helped. This was how to set up the dihybrid cross. After you've set it up, how to calculate or find the ratios for the genotype and the phenotype. All right. It gets a lot more tricky when both parents are heterozygous. That's when you see a lot more variation in genotype and phenotype, okay? So if you want the extra challenge, do this again at home, but do it with 2 parents that are heterozygous for both traits, all right? Cool. I hope that this was helpful. I hope that you guys learned something, and I hope that you didn't find this too dreadful, it being my first time creating a screencast for you like this. So they will get better, I promise, as the year progresses. Thanks, guys. I will see you derps tomorrow.